The forgotten power of a single tree. Multiply your favorite plant endlessly and watch it grow beyond limits. No seed packets, no nursery visits, no price tags hanging from plastic pots. Just one living tree, quietly producing copy after copy of itself. For centuries, gardeners and farmers did exactly this. Entire landscapes were cloned from a single parent plant, using nothing more than water, time, and an understanding of how plants behave when they are wounded. One tree could become many, many could become forests. This was not folklore or luck. It was observation, repeated year after year and passed down not in books, but in practice. Yet somewhere along the way, this knowledge nearly vanished. Today, most people reach for a small jar of white powder instead. The question is not whether that powder works. The question is why we stopped trusting a system that allowed one tree to create infinite plants, and why we replaced it with chemicals sold on store shelves. A scene nature repeats without human help. A willow tree stands tall, flowing with the river's edge. A storm passes through, wind snapping one of its branches. The branch tumbles into the water, drifts downstream, and settles into wet soil. There is no gardener watching, no human intervention. Days pass, and something remarkable happens. From the broken end of the branch, small white roots begin to emerge. The branch does not rot. It does not wither. It anchors itself, draws water, and begins the slow transformation into a new tree. This scene plays out naturally across the world, in ditches, floodplains, and wetlands wherever willows grow. Damage does not end the tree's life. Damage multiplies it. What looks like destruction becomes reproduction. The chemical command, hidden inside the branch. This ability is not magic, it is chemistry. Inside young willow shoots flows a powerful plant hormone called indole-3-butyric acid, known to scientists as IBA. This molecule is a messenger. When a branch is cut or broken, IBA floods the wound site and delivers a clear instruction to the surrounding cells. Ordinary plant cells are told to stop acting like stem tissue and start acting like roots. The cells reorganize themselves, forming new structures designed to absorb water and nutrients. Root hairs develop. Vascular tissue redirects. The plant does not guess. It responds to a chemical command that has evolved for survival. This same biological signal is what modern rooting powders attempt to imitate. In willow trees, it is already present, flowing naturally through young, flexible shoots. That is why even a pencil-thin twig pushed into damp earth can grow into a full tree. Why rooting alone is not enough. Root formation alone, however, is not enough. A cutting is extremely vulnerable. The moment a branch is cut, it becomes an open wound. Bacteria, fungi, and rot are immediate threats. Many cuttings fail not because they cannot produce roots, but because they die before roots have time to form. Willows solved this problem long before humans understood it. Alongside rooting hormones, young willow tissue contains compounds that convert into salicylic acid. In plants, salicylic acid functions as an immune signal. When tissue is damaged, it triggers a defensive response. Cell walls strengthen. Protective proteins are produced. The spread of pathogens is slowed. Modern laboratory studies later confirmed that salicylic acid reduces the risk of fungal infections and damping off disease during the earliest stages of plant propagation. This means willow cuttings are not only encouraged to grow roots faster, but are also protected during the most dangerous period of their survival. Growth and defense are combined in a single natural system. How gardeners knew long before scientists. Gardeners noticed these effects centuries before hormones had names. Through observation, patterns became clear. Some plants rooted easily, others failed repeatedly. Whenever willow branches were involved, success rates improved. People experimented quietly. Cuttings were soaked in water that had held fresh willow twigs. Plants were watered with simple infusions made from young shoots. Over time, experience taught an important lesson. The youngest, most flexible willow growth worked best. Fresh green shoots contained the highest concentration of the chemicals responsible for rooting and defense. Older woody branches were less effective. None of this required microscopes or textbooks. It required patience and attention. Knowledge passed from gardener to gardener, not through patents or packaging, but through results. The rose experiments that changed minds. By the mid-20th century, this traditional practice began to enter formal testing. In the 1960s, a bulletin circulated within the American Rose Society describing a simple experiment. Rose cuttings were divided into groups. Some were soaked in plain water. Others were treated with commercial rooting powders available at the time. A third group was soaked in willow water, prepared by steeping young green willow twigs in water. The results surprised even experienced growers. The cuttings treated with willow water rooted more quickly, produced more extensive root systems, and survived at higher rates than those treated with commercial products. In some cases, rooting success exceeded 90%. Even rose varieties considered difficult to propagate responded. For many gardeners, this was not theory. It was proof that an old method could outperform modern alternatives under the right conditions. Where willow water reaches its limits. Willow water was not a miracle solution for every plant. Controlled trials revealed clear limitations. Easy-to-root species such as apple often showed little difference regardless of treatment. More difficult species produced mixed results. Low concentrations of willow extract could improve survival, while high concentrations could be toxic and kill cuttings outright. Effectiveness depended on multiple factors, including the species of willow used, the age of the twigs, the season in which they were harvested, and the method of extraction. Willow water worked best with softwood and semi-hardwood cuttings, young stems already inclined to root. Hardwood cuttings and species resistant to propagation showed little benefit. This variability would soon matter greatly. The moment science took control. In 1935, researchers Percy Zimmerman and Alfred Hitchcock at the Boyce-Thompson Institute in New York set out to isolate the chemical responsible for reliable root formation. Their goal was not to preserve tradition, but to achieve consistency. They tested pure compounds one by one, observing their effects on plant cuttings. Their breakthrough came with synthetic indole-3 butyric acid. Unlike natural extracts, synthetic IBA proved stable, measurable, and effective across a wide range of plant species. The discovery earned them a prestigious scientific award and marked a turning point in horticulture. Rooting could now be controlled with precision. Nature had competition. From laboratory discovery to store shelves, science quickly attracted industry. Within a few years, chemical companies secured patents to manufacture synthetic IBA at scale. By the early 1940s, products such as Hormidin appeared on store shelves. These products were marketed as scientific progress. They were shelf-stable, easy to use, and delivered a measured dose every time. The message was simple and compelling. No need to forage for willow twigs. No need to wait for the right season. No need to experiment. For the first time, a plant's natural rooting signal was packaged, branded, and sold. What once flowed freely from a tree now passed through patent offices and retail catalogs. The economic reason we switched. The shift from natural methods to commercial products was not driven by performance alone. It was driven by economics. Willow water is powerful, but it does not last. The active hormones and defensive compounds degrade over time. Microbes grow. Within days or weeks, the solution loses its effectiveness. Synthetic powders, by contrast, remain stable for years. They can be shipped, stored, and sold repeatedly. One willow tree can supply a neighborhood for free. A small jar of powder generates profit over and over again. Retail systems favor predictability and repeat sales, not abundance. As buying became easier than making, knowledge that was no longer practiced began to fade. How willow water is still made today. Despite this shift, the natural method never stopped working. Preparing willow water remains simple. Young, pencil-thin green twigs are collected, preferably in spring or early summer, when hormone concentrations are highest. Leaves are removed, and the twigs are cut into small pieces to increase surface area. These pieces are then steeped in water, either hot or cold. Hot water extracts compounds more quickly, producing a pale infusion within 24 to 48 hours. Cold water takes longer, usually 2 to 3 days, but can be just as effective. Once strained, the liquid is used immediately. Cuttings are soaked in the solution for several hours, allowing the natural hormones and salicylic compounds to penetrate the tissue. After planting, the same solution can be used for early watering, delivering repeated doses during the most vulnerable stage of growth. Why shelf life changed everything. There are limits. Willow water is a living solution. Microbial growth begins quickly. At room temperature, it can turn cloudy and sour within a week. Refrigeration slows the process, extending its usefulness to a few weeks. Freezing can preserve it longer, locking in the active compounds until needed. These limitations explain why willow water never became a retail product. Freshness is essential to its power, and freshness does not fit well on store shelves. Synthetic powders, while lacking the full complexity of natural plant chemistry, offer convenience and longevity. The knowledge is returning. Today, plant propagation remains shaped by what can be packaged and sold, yet knowledge is resurfacing. Gardeners are once again experimenting, comparing natural methods with commercial products, running side-by-side tests, and measuring results instead of trusting labels. What they are rediscovering is not that chemicals are useless, but that they were never the only option. Nature's system was not replaced because it failed. It was replaced because it was inconvenient. What will we choose? One tree really can create infinite plants. That truth has never changed. The chemistry that makes it possible still flows through young shoots and flexible stems. The experiments that proved it still stand. What changed was our relationship with that knowledge. We moved from observation to consumption, from participation to purchase. The question is no longer whether chemical rooting products work. The real question is whether we want growth that depends entirely on something we buy, or growth that begins with understanding how nature already works. Abundance does not come from a bottle. It still grows wild, along riverbanks and in broken branches, waiting for those willing to remember what one tree has always been capable of creating. If this video helped you see nature differently, like, share, and subscribe for more stories that dig beneath the surface.